Hi everyone, and welcome to this week's lab where we will be looking at neurons and the effects of different drugs on heart rate. So same thing as every week, you guys are going to watch some videos and collect some data and carry out this experiment. You'll write your lab summary, upload it to Canvas or your Flipgrid, and then instead of a research proposal, that is still just for the in-person labs. Instead of the research proposal, you guys are going to be adding that experimental design section to the beginning of your labs. So I would recommend doing the experimental design before you start the lab. The whole point is to think through it and have an easier time processing what's going on in front of you rather than just trying to rush through and get it done as quick as possible. So we're going to go over some helpful tips for looking at the heart rates. This is a very vague question. Please don't just copy and paste this and put this into your lab summary because we are looking at heart rates being different than others, but we're looking at the cause and what's actually causing those heart rates to be different. And so we'll look into that this week. So just for some background knowledge, we're going to be thinking about how heart rate is regulated, and a lot of this has to do with these neurons. So the signal from these neurons are going to come in from the dendrites. They'll be processed in the cell body, whether or not it sends an actual signal. It'll travel down the axon, and then at the end of the axon, at these terminals, you'll get to the synapse, which the synapse is just the fancy name for the gap in between the two cells. And so at the synapse, once this signal comes down the neuron, it will signal the cell to release these little packets of neurotransmitters that they're called vesicles. And so they will release that neurotransmitter into the gap. And so at this gap, these neurotransmitters, these are just, they can be very specialized cells like serotonin or anything like that, or it can just be an ion. But in any case, they will bind to receptors on the other side. So there's a couple words that you're going to need to keep straight here when you think about it. So the vesicle is the packet itself. The neurotransmitter is what's contained in that vesicle, and neuro meaning, brain or neuron, transmitter means it's transmitting the signal, and then it'll bind to a receptor. And so the receptor is receiving that signal and causing something to happen in that postsynaptic cell, or the cell that comes after the synapse. And in this case, we're thinking about muscle cells, and specifically the muscle cells in the heart. And so these, depending on the neurotransmitter that's released, will change what response happens in the postsynaptic muscle cell, or the heart. So the drugs that we use today, they can mimic a certain neurotransmitter that may cause a response. They can block the receptors, or block the neurotransmitter from binding to receptors, or they may just have no effect. And so you can think of these as having different effects on the heart rate. So if they're mimicking a neurotransmitter that may increase the heart rate, then adding that drug should increase the heart rate. Now, if the heart would normally be beating at a certain rate, and then you block those neurotransmitters that would be causing it to beat at that rate, then you might slow down that heart rate. Or if there's no effect, then you're obviously not going to see any effect. And so those are just a couple examples of many different ways that you can think about this happening. And I really want you to think about what possible effects these could be having. Brainstorm, could it be mimicking something that actually causes it to slow down? Is it mimicking something that is causing it to speed up, or, stuff like that. So what effect do you think the following drugs will have? We're going to be looking at nicotine, which a lot of people, their first thought is going to be cigarettes. And so you can think about what kind of effect would nicotine have on the heart rate. We're going to be looking at ethanol, which is the chemical that causes alcohol to be a depressant. And we're going to lastly look at caffeine, which I'm sure a lot of us are very familiar with how caffeine works. So think about what effect you would expect caffeine to have on the heart. I know I say the same thing every week, but this is stuff that's important for the in-person labs. This will be talking about, the procedures that you'll be following. Take a look, read the lab protocol, and just understand what we're doing in the lab so that when you go to write your lab summary, you know what's going on. The organisms we'll be looking at this week are aquatic organisms called Daphnia. They're found a lot of places, and their physiology is pretty easy to manipulate, so they're going to be good model organisms, and you'll be able to observe these different effects. We obviously don't want the Daphnia swimming around when we're trying to watch them, so this is just the method of keeping them in place. You guys will be watching a recording of the Daphnia in the microscope. It was on low power and it took a while to focus, and so that's how it was done. So when you're looking at the Daphnia, you guys are going to be watching a recording and collecting your own data on the change in heart rate. So let's make sure that we are able to identify these different parts of the Daphnia so you're not counting the wrong movements. The section labeled A are the gills, and a lot of students will look at the gills and see that they're moving and then just get a completely wrong heart rate. This dark part is the eye spot. It's not going to be moving a ton. These are obviously antennae. I hope you wouldn't confuse those with the heart. This translucent section is the heart. It'll be beating, and E is while it looks like it could be the heart because it's large and dark, but it's actually a baby Daphnia. And so there may be some of those in these videos as well. But make sure that when you're looking at this, that tiny translucent part labeled D is what you're counting the beats of. So you guys are going to get a couple of videos with the different chemicals where you're going to have to go through and record the B4 heart rate for the Daphnia. There'll be a gap and then the chemical exposure, and then you'll come back and you'll record the same Daphnia heart rate after adding the chemical. And so why is it done this way rather than taking a group of Daphnia beforehand and then you don't add the drug and then you have a group of Daphnia where you do add the drug? And so because these are living organisms, there can be a lot of variability in just the basal heart rate. And so by taking one individual Daphnia and adding the chemical and just seeing what its base is and then seeing what its heart rate is after adding the chemical, each Daphnia can act as its own control. And so this is a really important method, and so try and keep this in mind when you're writing it up. So you guys are going to be using a data set that already has some data in it. This is because in the in-person labs, each lab group would be sharing their data with the rest of the class. And so there will be 3 trials already recorded, and the data that you record from the videos will be the 4th trial. And so you'll need to make your graphs using these data, and you're going to really be looking at the change in heart rate, not just which ones start lower or which ones start higher. Make sure that you're looking at that delta, or the pre-exposure versus the post-exposure, because that's really what we're looking at, is how did it change. And so if you have any more questions, please feel free to send me an email or pop into office hours, and good luck this week.